did mention that once they've elucidated the structure, they also postulated that there must be some way of copying the genetic material so that it can be passed on to subsequent generations. So, that is what today's lecture is going to be on, with a little bit of mutations at the end of it. And so the learning objectives are to understand the process, though I'm sure most of you probably already know it, so it's more likely to reinforce the concept. Have an appreciation for the experiment that led to the discovery of DNA replication. Understand in a bit more detail maybe than what you've already had before you came here about DNA replication. And then we'll just touch on mutations and the importance of DNA repair. At any point as I'm going through, if you need me to slow down or repeat anything because you're taking notes, then by all means just shout out and let me know. So this is the basic concept of DNA replication, and it's as you already know. You've got your DNA double helix, which unwinds, and then sometimes these are called the parental strands, act as a template for the synthesis of new, sometimes called daughter strands. And it's called semi-conservative replication. And it's what we know is how DNA replicates itself. But at the same time as semi-conservative replication was proposed, there was also two other models. Some may know the conservative model, but there was also another model that was proposed at the time as well, and this was called the dispersive model. So, conservative model, as the name suggests, so this isn't really rocket science at all, is that somehow the parental strand will unwind, act as a template for the synthesis of a new strand, but then the original parental strand will reform, giving rise to one conserved original strand, and then a completely new formed daughter strand. And then in subsequent rounds of replication, you can see what's happening. So that was one other option to the semi-conservative model. And then there's also this dispersive one, where basically what was postulated was that the DNA helix stayed as a helix and then got fragmented up and still acted as a template for the synthesis of new strands or daughter molecules, and then somehow it was reassembled all together so that you had, as the name suggests, parental DNA interspersed or dispersed with the new daughter strands as well. So they were the three models that were proposed at the time. And this was round about in 1953 after Watson and Crick had elucidated the structure. So it wasn't until 1958, so a good few years later, that Messelson and Stahl devised an experiment to determine which model, if any, was responsible for DNA replication. These guys are still alive today, 89 and 90 years old. So this is the experiment, quite simplistic in its setup. And the important part of the experiment was on the basis of nitrogen labelling. So as we all know, and as the structure was elucidated, DNA contains nitrogen, it's a major component of DNA. And nitrogen exists in two forms. So it exists in a light form, which is the most common form of nitrogen, and it also exists in a rare, heavy form called 15N. Okay, so there's the two forms of nitrogen: heavy nitrogen and then a light nitrogen. Okay, so what they did, they took bacteria, they took E. coli, and they grew it in culture media containing only 15N, so only the heavy form of the nitrogen. So they did this until, for several sorts of rounds of cell division, to ensure that they had a bacterial cell population that when they've gone through replication, and had made new DNA strands. The new DNA strands and then subsequent new population contained only the heavy form of the nitrogen. Okay? Is that all right with everybody? And then what they decided to do was take a small section of the bacterial population. They burst the cells, they lysed the bacteria so that they released all their content. They extracted the DNA, and then they added it to a cesium chloride solution, which forms on centrifugation a concentration gradient. When solutions are quite dense, they would sediment to the bottom of the test tube. Light solutions or samples would centrifuge closer to the top, and then obviously intermediate of density would be in the middle. Okay? So what they did was, after culturing the bacteria for several rounds of cell division, so that they had a population that only contained 15N, and then transferred it into they transferred the bacteria into a solution containing only the light version. Bacteria, and especially E. coli, if they're grown in the lab with optimum nutrients and temperature, can divide every 20 minutes. Hence the reason why, after one round of replication, they took the sample, lysed the bacteria, extracted the DNA, and centrifuged it in a solution of cesium chloride. And so this is what they hypothesized. So if the actual conservative model was the correct model of replication, you should have two bands. One in which the DNA is of a heavy density because it's the conserved parental molecule and is only containing the heavy nitrogen. Or there should be another band where, after being grown in the light form, then the new DNA strands will contain the light nitrogen form. So there should be two bands. The semi-conservative model and the dispersive model both lend themselves to being intermediate because they contain a mixture of both heavy and light nitrogen. Everybody okay with that? Yeah, I know it's late in the evening. So this is what they observed, which is in fact a sample which is in the middle, so meaning it's intermediate. So straight away then, you can discount the conservative model because there's no heavy nitrogen to argue that there's any of the parental DNA conserved. Okay? That was only going to sediment to the bottom. But that then didn't mean that we could distinguish between semi-conservative and dispersive. So they went through a second round of replication. What they did there, so it's still being grown in the light nitrogen-containing medium, extracted the DNA, etc., did exactly the same thing, and then what they found was that there was still some intermediate sample, and then also they found that there was a light segment as well. Okay, so that showed then, obviously this conservative was still discounted, but this also showed then that the dispersive was discounted, because if it's dispersive, then you still only get one intermediate band. Okay, but what it's showing is that when the semi-conservative replicated, so the DNA helix split apart, you would get then some of this original molecule, and then you'll get the new synthesized DNA where it's completely got the light form of nitrogen incorporated into it. Is that okay with everybody? Yes? Nodding overhead, I'm liking that. Okay, so this might just help to explain a little bit better, where you can see the sort of usual notations that we use for DNA, where you've got your lines. So you've got the original parental molecule, or parental DNA, that's labelled with a heavy nitrogen, and then when it's spun down, in the centrifuge, it sediments further to the bottom. Then, as you can see here, pulls apart, you get then parental DNA forming the template for which synthesis of new daughter strands happen, but these new daughter strands now contain the light form of nitrogen. Okay, so that forms here at 100%, intermediate, and then through subsequent cell divisions, you can see then that eventually the heavy solution, heavy nitrogen, is being diluted out, and so then there's less and less of the band sedimenting in the middle. Okay? Is that all right with everybody? So, just to remind you that replication takes place in the S phase. And then, where does replication occur? Okay? It's all well and good knowing that there's a structure of the DNA. It's all well and good knowing that it does replicate. But where in the DNA does it replicate? So, it actually replicates at the origin. So, no shit. But, in science, sometimes it's not that straightforward and they come up with some really weird names, okay? But for once, we have an origin of replication. But even sort of like today, we still don't quite know sometimes where the origin of replication will occur. Even with all the research that's being done and the new techniques that we have, we can't categorically say that there is a conserved sequence or a pattern which you can significantly say that is the origin of replication. So even with next generation sequencing, we still are not sure. So there are some sort of hypotheses to say that in prokaryotes, sequences that are very rich in adenine and thymine are seen as the origins of replication. Why would that be? Does anybody know? Can anybody hazard a guess? Is anybody thinking, or are you all just asleep, or waiting for Junior Baker to start or something? Does anybody want to say anything? Yeah, it's to do with the fact that they've got less hydrogen bonds. Okay, so that's what the argument was there. Adenine and thymine are only bonded together by 2 hydrogen bonds, so it might be easier for them to pull apart, and the bacteria don't have to expend so much energy. It was thought the same as well for eukaryotes, but with more and more research being done, they're actually thinking that origins of replication for multicellular organisms are rich in guanine and cytosine. Something to do with the structure in which the DNA is folded allows better access for all the different proteins involved. But as I said, we're still not 100% sure. So what we do know is that replication occurs at the origin of replication, and when the DNA helix unwinds, we get what's called a replication bubble. This replication bubble then has what's called replication forks at either side of the replication bubble. Replication occurs in both directions. So, as the name suggests, replication occurs bidirectionally until the whole molecule is copied. In prokaryotes, they don't have such a large genome, and so there's usually only one, or there is only one origin of replication. In eukaryotes, however, due to the size of our genome, we have tens of thousands of origins of replication all occurring at the same time to make sure that our DNA is replicated in a feasible amount of time. So you can see, if you have a look here, there are thousands of these occurring at the same time, and eventually these replication bubbles will fuse until you get the daughter molecules. And if you just have a look on the scanning electron micrograph here, you'll see that there's 2 replication bubbles very close together, and eventually they fuse. And this naff little gif that I tried to, I didn't make it by the way, if that was to continue, then you see that eventually they get bigger and bigger until obviously then they come into the 2 strands. So it's been hypothesized as well that the rate of replication in prokaryotes occurs at around 1,000 nucleotides a second. And for eukaryotes, it's only 50 nucleotides a second. So the argument for us having a slower replication is to ensure that we have great accuracy and fidelity of the DNA that's being replicated. Even so, it takes about 8 hours to completely replicate our whole DNA. It's even been postulated that if we only had one origin of replication, it would take us around about 35 days just to replicate one chromosome. So that's just to put it in context, why we need so many replication origins. Is that okay with everybody? And my amazing facts? Right, so, not new to you, here we have a nucleotide, and we've got our sugar, our base, and then our phosphate groups, and the nucleotide usually will only have one phosphate. So it has 3 here. Cause. Synthesis of a new strand and nucleotide incorporation. So each nucleotide is made up of a nucleotide triphosphate. And what you'll notice as well in this diagram is that we have orientation assigned to the DNA strands. So you've got your template strand, so your parental strand here, going in the 3' to 5' orientation, and then you have your growing strand in the 5' to 3'. I'm sure most of you know what the primes stand for, but just in case you don't, and you don't want to shout out, it's to do with the number of carbons that are in the sugar. Okay, so we have 1, 2, 3, 4. The 5th carbon is attached to the 4th, and on the 5th carbon is where the phosphate groups are attached, and on the 3rd carbon we have a hydroxyl group. The enzyme DNA polymerase is what is responsible for incorporating nucleotides into the DNA chain, and it can only do it in a 5' to 3' direction. Okay? And that is really important to remember and to understand, because it forms the basis of a lot of molecular techniques, and obviously it is just nature as well. Okay? So it's called 5' to 3' because the 5' end containing the phosphate are added to the 3' hydroxyl group in the growing chain. When DNA polymerase does add this, we lose 2 phosphates so that you only have 1 phosphate in the DNA backbone. Okay, is that all right with everybody? Remember 5' to 3', and that DNA polymerase only attaches in 5' to 3'. The other thing as well is that DNA polymerase cannot synthesize a new DNA strand from scratch. Okay? It can only attach nucleotides to a pre-existing strand. So how do we kind of overcome this problem? We have what is called RNA priming. There is no DNA polymerase that's been identified that can synthesize DNA from scratch, but RNA doesn't have that problem. Okay? RNA can be synthesized from scratch without needing a primer. So what we have, we've got your template DNA to act for the DNA synthesis. The enzyme called primase will come along and it will synthesize what's called a primer of around about 5 to 10 nucleotides. This then allows there to be a free 3' hydroxyl group to which then DNA polymerase can come along and start adding nucleotides and start to elongate then the DNA strand. So we'll only look at bacterial replication, prokaryotes and eukaryotes are very similar. More work has been done on bacteria, but they are very similar. But for simplicity, I will stick with bacteria. So if we take the replication bubble, okay, and we just look at the top strand, you can see that the enzyme helicase has come along and it's unwound the DNA, parental strand, into two single strands, sorry, at the origin of replication. Alright? We also have what are called single-stranded binding proteins. So they attach as soon as the DNA helix is unwound to ensure that the single strands do not reanneal and form the helix again. So at the origin of replication, as we said, there's no ability for DNA polymerase to come straight along. So you have your primase that can synthesize RNA primer of, like I said, 10 nucleotides, and then you've got DNA polymerase coming in and taking over and elongating the strand. This is called the leading strand and is made in a continuous fashion all along the line. Okay? As helicase and what keeps unwinding the DNA double strand, DNA polymerase can just come along and keep inserting new nucleotides as a new template is being exposed. It only requires one RNA primer as well, and then it's synthesized in a continuous fashion. However, on the bottom strand, that is not the case. On the bottom strand, you will see that the new strand has to be synthesized in fragments. And this is because it has to work away from the replication fork. So unlike the top strand where it can work, it's working towards the replication fork in a 5' to 3' direction, the bottom strand cannot. And it's called a lagging strand. The lagging strand is so called, they're made at the same time, but because they have to work away from the replication fork in a 5' to 3' direction, you have to wait for enough nucleotides to be exposed by the helicase before synthesis can occur. So it lags behind, not significantly, but it lags behind the leading strand. And what you'll see as well then is that each time there is nucleotides exposed on the bottom strand, you need a new RNA primer. Okay, so as more is exposed, you have to have an RNA primer. So you could say, for example, we started down here and we had unraveled the DNA helix, got your RNA primer, and then synthesis occurs. This bit then, it has not been exposed. It becomes exposed to show the nucleotides. A new RNA primer needs to come along because the DNA polymerase cannot add in this direction. Okay, it cannot add in 3' to 5. Is that okay with everybody? Does everybody know what these fragments are called?
Okazaki.
Yes. Okay. Okazaki fragments, after the name of the Japanese biologist who worked out that both strands are not synthesized in a continuous fashion. And sometimes it helps people to think of the analogy of like a zip. You literally are unzipping your DNA with the enzyme helicase, and just like the teeth on your zip, if you pull it apart, you're starting to expose what classed as the teeth on your zip. They constitute in the analogy as your nucleotides, and as you pull your zip down, more and more of these teeth are being exposed to act as the template to which then DNA polymerase can bind. I don't suggest you start zipping genes or anything now, just to check, but trust me, it's. Okay? So here are a couple of important enzymes that are involved in DNA replication. I'm not going to go through them all now. Oh, I should say actually, yes, the bottom bits. Obviously, we do not have a DNA and RNA hybrid in our genetic material. So these RNA primers need to be removed. They are removed by DNA polymerase 1, and in doing so then, DNA polymerase 1 removes them and then elongates what's left over to join up the gaps. You'll also find as well then that once the new nucleotides are put in, after the primer has been removed, if you see down a bit by here, there is a small little gap in the phosphated backbone, and the enzyme DNA ligase comes along and is able to fill in that and prevent any gaps from occurring. Is that okay with everybody with regards to DNA replication? Yeah? Probably not really anything majorly new there for you to understand, but just digging into you in slightly a bit more detail. And there are enzymes. Oh. So just to consolidate it if it does work.
DNA polymerase enzyme adds DNA nucleotides to this RNA primer. Here is a closer view of this process within DNA polymerase. DNA polymerase aids in the process of adding complementary nucleotides to the template strand. First, hydrogen bonds form between complementary bases. Next, DNA polymerase facilitates the bonding between the phosphates and deoxyribose sugars, connecting the nucleotides of the new strand. DNA polymerase can only join nucleotides in the 5' to 3' direction. On the leading strand, the DNA polymerase continues to move towards helicase. On the lagging strand, this process must occur in small 5' to 3' segments as more lagging template is exposed. This representation of lagging strand synthesis has been simplified for clarity. Again, primase must lay a primer before DNA polymerase can continue the growing strand. These segments are called Okazaki fragments. On both new strands, a nucleotide replacement enzyme replaces primer with the correct DNA nucleotides. While the DNA polymerase continues along the leading strand uninterrupted, gaps are created on the lagging strand between each Okazaki fragment. DNA ligase seals the gaps and connects the fragments into a continuous strand. Helicase continues to unwind the original DNA strands. Both strands are replicated at the same time. The leading strand replicates continuously toward the replication fork, while the lagging strand replicates in fragments away from the replication fork. This process continues in multiple locations until the entire chromosome is replicated.
What I will say is that I don't really understand why they call it a nucleotide replacement enzyme. We do know that it's DNA polymerase 1. So, but other than that, sometimes I quite like to put these videos in so that you can see it in a more sort of 3D context. So that's DNA replication, but I think the last sort of 10 or 15 minutes we'll just talk about DNA repair, because it does go hand in hand. Because DNA replication is not 100% accurate. DNA polymerase do make a significant number of mistakes when it's incorporating the nucleotides, because nothing is foolproof. The DNA polymerase does have a proofreading ability. So it's able to check that it's incorporated the correct nucleotide into the DNA strand against the template strand, and if then they're seen as a wrong base has been put in, it's got what's called 3' to 5' exonuclease activity. So it can actually go back and incorporate a new strand, but only that one base. Okay, sorry, not strand, base. All right? So it's got proofreading ability, and it's been hypothesized that DNA polymerase will, on average, make one error every 100,000 bases. So you might not think that's that much, but when you consider the size of our genome, it can turn out to be then a very significant number of mutations that are starting to be introduced. And that's just through DNA replication. We also have endogenous chemical processes going on in our body that naturally damage our DNA. And then obviously we've got exogenous damage as well through environmental stresses such as smoking, ultraviolet light, pollution, etc. So all of these then things taken into consideration means that our DNA is constantly under attack and needs to be repaired. So apart from proofreading of DNA polymerase, which only occurs in DNA replication, we do have DNA repair mechanisms. So we have mismatch repair, nucleotide excision repair, and also we have recombination repair. So here you can see where the different types of repair mechanisms come into play. So you can see, for example, that X-rays, hydroxyl radicals, for example, will form abnormal bases, single-strand breaks, may even have abasic sites, meaning that there's no actual base there. And then the process that repairs it is called base excision repair. UV damage can cause thymine dimers. So instead of thymine binding to adenine, they will bind to each other, forming sort of distortions in the DNA helix, which are repaired by nucleotide excision repair. Then we've got double-strand breaks, and then we've obviously got mismatches as well. DNA repair is actually a very highly complex process, so I'm not going to go into that, especially not for year one, but all you need to understand is that there are enzymes, multiple enzymes, that function is to sense and detect this DNA damage. Once it's detected, they excise the damaged DNA and sometimes maybe some DNA around the sides of it, and then they use a template DNA on the other non-damaged side to then incorporate complementary base pairs to repair it. But that is a very simplistic overview, and depending on if I can get this to work. It sounds better than what it was.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in a partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA pattern. Double strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when the sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same.
You get this. That's to say that our DNA repair systems work, and on the whole they normally do. But we also can sometimes have errors and mutations in our DNA repair. And as a result of that, we have sometimes hereditary DNA repair disorders. So I'm only going to give you two, and it's not going to be tested on or anything like that in your continuous assessments, but it's not all about just getting your marks. It's about understanding the genetic components of everything. So just out of interest, I thought I'd give you two, one of which is called xeroderma pigmentosa, and as you can see here, it says that it's a hypersensitivity to sunlight. So as we saw in both the little cartoon and in the other slide, ultraviolet light damage causes thymine dimers, and it's usually repaired by the nucleotide excision repair pathways. However, there's mutations, or several, I think 9 or 10 mutations known to occur in the nucleotide excision repair pathways, which mean then that individuals that have these mutations are unable to repair their damage that they're exposed to. So they can't go out in sunlight, and if they do, they have to wear this type of clothing to protect them. But what you find is that as time goes on, they will still, you can't be completely shut away in the dark, even sort of like being at home, and through windows they'll be exposed. And so eventually, as time progresses, they get more and more DNA damage, and they obviously can't repair it. So it's really, really sad. Obviously, it leads to high incidences of cancer, and cancer is usually the common cause for death. So it says here life expectancy is around 29 to 37 years, but usually most individuals with XP don't live past the age of 20. So it just goes to show how much our DNA repair pathways are important to us. And then the other one that I just want to mention, which is one of the classic examples that I've given as well, is to do with Werner syndrome. So Werner syndrome arises as a result of mutations in the WRN gene. There's been about 60 mutations known to affect this particular gene. And as you can see, it gives rise to premature aging and retarded growth. Normal growth and development usually occurs up until puberty, and average age of diagnosis is around about 24. So the Werner gene, WRN gene, is involved in DNA replication and repair, as you would expect. And so when this is not functioning, mutations are accumulating and they're unable to be repaired. And as you can see with this individual here, at 15 years of age, she's looking as any 15-year-old would look. And then at 48 years of age, you can see the significant deterioration that's occurred as a result of not having these DNA repair enzymes functioning properly. So it's associated with grey hair, there's cataracts, osteoporosis, and then usually death occurs as a result of cancer as well. So, but just to sort of bring it home to the sort of medical implications that can happen as a result, and really sort of bring it into context, out of the textbook and into the clinic, is what you can see. But these are rare disorders, but nevertheless, obviously, they still do happen. So just to touch on the last couple of minutes, to just go into a little bit more detail about mutations. We have different types of mutations, and we have different types of DNA damage. So we have genomic instability in many different ways, but mutations are the most common ones that are known, and they are categorized in several ways based on the nature of the change. So we have base substitutions, base insertions, deletions, and then as a consequence of a change, so missense or frame shift. Here, normal is denoted in scientific terminology as wild type, okay? And then as you can see, substitution goes to feast because you substituted B to egg, and insertion gives rise to something completely different such as breast, and then deletion, best. So that's just a very cartoon analogy of how these different types of mutations from the wild type normal can then give rise to a whole host of different variations, and then the different types of consequences that we get. So I'm obviously going to go into the central dogma with you in the next lecture, where we go DNA, RNA, and protein, but I'm sure you already know about that anyway, enough for me to just show you that base substitutions, as the name says, is where one base is substituted for another, and sometimes they have absolutely no effect at all and are called silent mutations, because this base substitution doesn't change the amino acid from the wild type, so it's fine, it doesn't have any effect. You can also have missense mutations, and sometimes these don't have any sort of dramatic effect either, because although it's changed the amino acid from glycine to serine, it could be that it's not affecting the way that the protein folds, it's not affecting its shape, and then it's not affecting its structure because it might not be in a very important part of the protein. So again, they might not be that detrimental. But what is detrimental is if you have an alteration which causes translation to be stopped prematurely, and that could mean then that either a non-functional protein is made or no protein is made at all. And then base pair insertion or deletion. These are far more detrimental than the base substitutions. So as you can see, as the name suggests, it's insertions. So a base is inserted by mistake, or a base is deleted, and these can have significant downstream effects as to where the mutation occurs, because then what's called frame is completely out of alignment, and then where the mutation occurs, all downstream amino acids are not going to be correct. All right? So these do cause far more significant errors than just the substitutions. But we'll talk about that more in the next lecture. So that's just to give you an overview. But as the little cartoon said as well, mutations are not always bad. Mutations give rise to variation, and as we know, variation drives evolution. And in populations, we have what's called polymorphisms. So genes can exist without any sort of detrimental impact in different forms. Okay? So that's what polymorphism means, many forms. And they are alternative forms of a gene. And the most common one is when you have an alteration in just a single nucleotide, and these are called single nucleotide polymorphisms, or SNPs for short. And two common examples are the ABO blood group, and as you can imagine, you've got three different forms of the gene. It's either A, B, or O. And then we've got cytochrome P450 enzymes. They're involved in metabolism. Many, many different types of genes. Variations can occur in these genes. And even though the enzyme might still look structurally the same and function relatively the same, these slight alterations mean that individuals may metabolize drugs differently. Okay, so that's where polymorphism come in. So if we've got genome variation and we talk about polymorphisms and we talk about mutations, how do we know if a variation is polymorphic or whether it's perceived as being a mutation? Does anybody know? No? Pardon?
How many people have that mutation?
Yes, exactly. It's the frequency of which these variations occur. All right? So you've got gene variation. These mutations are going to occur, or as we say, variations. But what distinguishes these variations from being polymorphic, or whether it's a mutation, is the frequency that it's in the population. So a variation has to occur greater than 1% in the population for it to be classed as polymorphic. If it's less than 1% of the population, it's classed as being a mutation. Is that okay with everybody? All right? So obviously variation is occurring all the time, but when it becomes polymorphic, it means that it's being maintained in the population at a quite high frequency, so greater than 1%. Less than 1%, and it's happening through chance, and it's not really being maintained as a beneficial or different version. So, to bring it to an end, because it's been a long day. So, seems ages ago that we talked about cell-associated replication, but that's what we started off at. And just to bear in mind that replication is subject to mistakes. These can be repaired by DNA repair mechanisms and DNA polymerase, proofreading ability, but nothing is foolproof. And as a result, we do get mutations, and we also get defaults in our DNA repair mechanisms, which go on to lead to clinical manifestations of disease. Any questions?